Hi everyone, today I am giving you information on the topic slit lamp instrumentation. So let's move to its introduction. Slit lamp biomicroscope was invented by the Gulstrand in 1911. So this was invented by the Gulstrand. He was a scientist in 1911. Optical instrument which emits a narrow slit-like but intense beam of light used to examine the interior part of the eye from the cornea to the interior part of the vitreous. So it is a kind of instrument. This is a kind of instrument. It emits a small narrow beam of light by which we can examine the parts of the eye from the cornea to the interior part of the vitreous. Cornea is the starting watch glass like structure of the eye through which the light passes till we can see the vitreous part. Modern slit lamp with accessories devices not only provides the magnified views of every part for the documentation and quantitative measurements. Nowadays the modern slit lamp is there through which the LED is provided. We look from this area and the sitting of the patient should be there. Whatever the examiner see from this is magnified to the LED image so that we can perceive the good images here, and we can also show it to the patient that what's happening in the eye. Most slit lamp provide the range of magnification from 6x to 40x. So this one is the magnification of the slit lamp. It varies from the 6x to the 40x. Okay. Next one is principle. A slit beam of very bright light produced by the lamp. Now a lamp is placed inside it through which a slit of the light is coming. Okay, this kind of slit is coming. We can change the shape and the size of the slit. This beam is focused onto the eye, which is then viewed under magnification with a microscope. This is focused onto the eye. We can focus that beam onto the eye, and then under a magnification one can perceive the image of the patient eye. Next one, system of the slit lamp. Now the slit lamp consists of three types of functions. First one is the observation system, next one is the illumination system, and the third one is the mechanical system. Okay, now the observation system consists of three things. First one is the objective lens, second one is the eyepiece lens, and third one is the prism. Illumination system consists of these things: light source, condenser lens system, light and other diagrams, filters, projection lens, reflecting mirror, and the prisms. Mechanical system consists of joystick arrangement, up and down arrangement, patient support arrangement, and fixation target. Next one, there are the two types of slit lamp biomicroscope which are available in the market. First one is the Zeiss slit lamp microscope, and next one is the Hagstreit slit lamp microscope. Okay, so these are the both. Okay, both have the same parts, but the difference between both of them is that one part is so the illumination system of this slit lamp is at the upper side, whereas the illumination system is inbuilt inside this. Okay, so this is the main difference between both of them. Otherwise the working is almost same. Next one is the difference. So the difference between both of them is in Zeiss type the light source is at the base of the instrument. Okay, so this one is the Zeiss type where the illumination system is at the base, and here the illumination system is at the top. This one is the Hagstreit and this one is the Zeiss model. Okay, next one, parts of the slit lamp. We have three parts of the slit lamp. First one is the observation system, illumination system, and the mechanical system. So let's see one by one. Observation system consists of objective lens, an eyepiece lens, and the prism. Okay, now this one is the observation system. If we can cut down the part of the slit lamp, so it consists of observation system. Let's see one by one. The objective lens consists of two plano-convex lenses. Together they provide a composite power of plus 22. Okay, now the objective lens is present here. Okay, this one is the objective lens which is present here, from where we can see the object. Next one is the eyepiece lens. The eyepiece has a lens of plus 10 diopter power. It provides a good stereopsis. The eyepiece tubes are converged at the angle of 10 to 15 degrees. Now this one is the eyepiece. Okay, this one is the eyepiece from where the examiner can see. It gives a good stereopsis because two binocular like structures are there. One eye is placed here and one eye is placed here, so the examiner will look the single eye of the patient through both of the binoculars. Magnification is adjusted here. We can also adjust the PD. PD is basically the pupillary distance. The distance between both of the pupils can adjust it through this area. Okay, it can move to 10 to 15 degrees according to our eyes, so that we can take a good stereopsis, okay, and examine the patient properly. Next one is the prism. Image formed by a slit lamp is inverted. To re-invert the image, a pair of prism is used in the observation system. Now in the observation system, inside the observation system, there is a pair of prisms. Okay, so the prisms first prism form the inverted image, and the second prism re-invert that image so that it can perceive by us as a right image. Two prisms are there. Next one is the illumination system. Illumination system means lighting system, from where the light comes. Now the illumination system consists of light source, condenser lens system, slit and other diagrams, filters, projecting lens, reflecting mirror or the prism. So let's see one by one. Okay, so this is the illumination system which is set up inside the slit lamp and it is not visible to us. Okay, so let's see. Light source. Initially an earth lamp was used as a light source, which was followed by the nitra lamp, arc lamp, mercury vapor lamp, and finally the halogen lamp. It provides the illumination of 20,000 to 40,000 lux. Okay, so it is basically the names of the lamps. Initially that lamp was used and now the halogen lamps are used, and this provide the illumination of 20,000 to 40,000 lux. Okay, here the lamp is used. This one is the lamp which is used for the illumination system. Okay, next one is the condenser lens system. It consists of a couple of plano convex lens with their convex surface in the opposition. Okay, these are the condensers in which the plano convex lenses are at the opposition from one another. Okay, next one, light and other diagrams and the filters. The height and the width of the slit can be varies according to the two knobs provided for the purpose. Now the knobs are provided here. Okay, for the vertical slit we can change it. We can also increase the width. For example, this one is the slit and we can change the width of this slit, and we can also change it horizontally as well as the vertically. How much thickness is required and how much size is required, everything get changed by using these two knobs. Next one is the filters. Different filters can be inserted into the illumination beam. Cobalt blue filter, red free filter are provided in most of the models. There are the different types of cobalt blue filter. It is used for seeing the epithelial defect. If the patient has the epithelial defect on the cornea, then we can see it by using the cobalt blue filter by staining with the fluorescent strip. And next, yellow filters is mostly used in this slit lamp. Next one is the green filter for examining the retina sometimes, and these kind of filters are present here for examining the different parts of the eye. Next one is the projection lens and the reflecting mirror or the prism. It forms a single image, the slit at the eye. The diameter of the projection lens is fairly small. Okay, so the image passes on through it. This one is the projection lens. Okay, image is formed on the projection lens and then there is a mirror present here. After the image passes on through the projection lens, it falls on the mirror, and then through the mirror it settles on the patient eye so that the examiner can look the patient's eye. Okay, so reflecting mirror or the prism. A prism or the mirror reflect the light along a horizontal axis. Okay, so that we can perceive a clear image of the patient. Next one is mechanical system. Now the mechanical system consists of the joystick arrangement, up and down movement arrangement, patient support system, and the fixation target. Okay, so let's see. This one is the slit lamp. Okay, joystick arrangement, up and down arrangement. Now the movement of the microscope illumination system is done by using this joystick. We can use this joystick to do up as well as down. Okay, according to the position of the patient. Here the patient will keep the chin rest, here the patient touches the forehead, and according to our size we can adjust the up and down by using the joystick. Next one is up and down arrangement. The up and down movement is obtained by the same sort of screw device that moves the whole illumination system viewing up and the down relative to the chin rest. Okay, we can do the up and down by using this area under a certain limit, but we can also do the up and down by using this. Okay, here the small knob is present. If we can rotate it, then we can do up as well as the down. Next one is patient support arrangement and the fixation target. Okay, now the patient support system is here. In some of the slit lamp, there is a part present here so that the patient can hold that part. Okay, for proper sitting. Here this one is shown. The patient can hold this, and comfortably the patient can sit here so that the examiner can examine the patient. Next one is fixation target. A movable fixation target is given. Okay, so this one is the knob present here which the patient have to hold, and this one is the fixation target. A light is present here. Red colored light starts blinking. If the patient is PL positive, the PL positive means the patient can only perceive the light rays. Okay, so we will give the target to the patient, then you have to look at this target. If the patient is PL positive from the one eye or both eyes, then we will ask the patient that look at that direction. So this one is the fixation target which we can give to the patient. This was the end. Thanks for your patience listening.